Hi, I'm Bill Rapisi, Executive Director of Lymphatic Education and Research Network, and I just want to take this opportunity to thank all of you for joining us today for our symposium series. I also want to take this opportunity to thank all of you who have become members of LE&RN and help support this series so that we can continue bringing this kind of groundbreaking information to you in your homes and offices. I would also suggest for all of you who have not become members of LE&RN, $5 a month, you can become a member of LE&RN and help support this programming and the research that we do. Thanks very much for joining us, and I look forward to having you become a member.
Hello, everyone. Welcome to this webcast. It's my pleasure to introduce the webcast on behalf of LE&RN. I believe many of the audience might already know me in one context or another. My name is Dr. Stan Rockson. I'm one of the founders of LE&RN and also the Alan and Tina Neal Professor of Lymphatic Medicine and Research at Stanford. I have the pleasure to welcome you today to this webcast, and perhaps we can look at the first slide, which will give you the title. We're going to be talking about clinical trials for lymphedema. So it's my pleasure to introduce Dr. Joanne Quan. She is the Chief Medical Officer for Eiger Biopharmaceuticals, located here in Palo Alto, California. Joanne and I have had the pleasure of working together over the last year or two in creating a very exciting new clinical trial for lymphedema. It's come to our attention that because there have been so few opportunities for lymphedema to be the benefit of clinical trials, it is the case that many of you who have lymphedema or are concerned about lymphedema are relatively less aware of the structure of clinical trials, why they're performed, why they are performed in the way in which they typically are undertaken. And we thought it would be really useful to the community to present some general information about clinical trials and then to talk specifically about the clinical trial that Eiger Biopharmaceuticals is sponsoring that is such an important advance for the field of lymphedema. So I'd like to turn things over to Joanne so she can talk a little bit about clinical trials.
Thanks, Stan. It's a pleasure to be here today. And as you mentioned, we've been working together for the past couple years. And I think you've helped me really appreciate the unmet needs that are in lymphedema and a lot of the suffering that patients experience these days. So I'm really hopeful that by working with you and working with other people in the lymphedema community, we can change the picture of that. We at Eiger have been developing an investigational medication called Ubedimix that we think has a lot of promise for treatment of lymphedema. But before we get to that, let me just share some general information about clinical trials. So I know that clinical trials are new for many of you in the lymphedema community. So I put together just a couple slides in terms of basics so that the user community can understand the types of clinical trials, and then we can talk more specifically about the trial that Dr. Rockson and I have been working on. So there are many types of clinical studies. One kind is an observational study. In this, you observe what happens. So this can be done to look carefully at natural history, to understand the disease across a variety of different patients and manifestations, understanding what happens over time, what happens with various comorbid conditions. So those are very important studies to help understand a disease and what happens over time. And I believe that Stanford has a registry, which is this kind of study. What we typically think of as clinical trials test something. So they test new ways to either prevent, detect, or treat disease. And the treatment may be a drug, a procedure, a device, or even a particular way of using existing drugs. And our trial actually tests an investigational drug for lymphedema. Next slide. So some basic terminology that we tend to use. Clinical trials are in three phases. Simplistically, phase 1, phase 2, and phase 3. Phase 1 is often the first trial, which looks at safety and identifies some side effects. And often these are what we call first-in-human type studies. For a drug that has never been in people, we're looking very carefully at a small number of people to see what side effects are identified. This kind of trial may include a range of doses. Oftentimes you start at a low dose and you escalate, and you look at what kind of exposure you get, carefully look at side effects, and evaluate as best you can. phase 1 trials are often done in healthy volunteers and often in a fairly limited number of patients. With that information in phase 1, then we proceed to what we call phase 2. And here, I think, is where we start to think very carefully about patients with different diseases and start to ask the question, does the drug look like it works, and is it appear to be safe? So we start to look at what we call safety and efficacy. Oftentimes in phase 2 trials, we're looking at a range of doses because we haven't identified specifically what dose may be appropriate. Phase 2 studies often may take into the teens of patients per study. Our study, for instance, what we call the Ultra study, is enrolling around 40 patients. So that's a very reasonable number, and sometimes it goes into the hundreds. With information from phase 2, assuming that the drug does look to be working and is safe, then we would proceed forward to phase 3. Okay. So for phase 3, we specifically are looking to characterize safety and efficacy. We look at large numbers of patients, and we are definitely looking at patients with defined diseases. Oftentimes, we need to look at fewer patients if it's a rare disease. FDA and other regulatory agencies recognize that it's harder to find a large number of patients with rare diseases. Next slide. Next slide. So one of the common questions that we often have is, how are patients protected in clinical trials? We're talking about drugs that are used in an investigational manner. So they're not approved by FDA in the sense that you can go to the drugstore and get it, and your physician cannot prescribe it. But there are a number of different levels of protection that you as a patient who's considering entering a trial should be aware about. All studies that are done are reviewed by what we call IRBs, or institutional review boards. In some countries, it's called an ethics committee. And it's a group of people with both scientific background and also with lay background. Lay people are members of IRBs as well, who provide the scientific and ethical oversight of trials. So we submit a detailed study plan that we call a protocol to an IRB or ethics committee, and they review it, and they come back to us with questions. Questions about, is this procedure appropriate? Does it make sense to put patients through this? What value is this? So we, as a company, and working with investigators like Dr. Rockson, carefully consider those comments and then come to agreement with them about what's a reasonable study plan to proceed with. Some studies may have what we call data safety monitoring boards, an independent group of physicians, perhaps a statistician, to look at the safety and advise us if there's any concerns. As well, in this country, the Food and Drug Administration, or the FDA, has oversight for all trials, looking at new medicines and devices, and also in that process approves all new drugs in order for them to be available. We as a company, when we conduct a clinical trial, need to file our study plan and actually a lot of the detailed background about both the disease and the drug that we're testing with the FDA, and the FDA has oversight over this. So they will be essentially looking over our shoulder to make sure that the patients are safe. And as well, part of the patient protections is written informed consent. For any of you that may have considered participating in a study, you probably at some point have been handed a rather long and detailed form which lists the purpose of the study, lists all the study procedures, lists information about the drug, any cost, what's known about the risks and benefits. And this is part of ensuring that all patients who are considering participating in a study really understand what the study entails, what is known about the drug, and what people hope to find. It's important to recognize that participation is always voluntary. So we want to make sure that everyone is informed and that people know what's involved. And even if you sign up for a study and you decide that you don't want to participate at some point in the future, as a patient, you have the right to withdraw at any time. Those things are always important parts of informed consent. Next slide. So there's actually a number of good resources that are out there about clinical trials. The NIH, National Institutes of Health, has a very nice website. If you just look on nih.gov and search clinical trials, you'll find some of the basic information that I summarized in the previous slides. This NHLBI is a little bit more direct way to get there. And also clinicaltrials.gov is really a clearinghouse listing of most of the trials that are done, both in the US and elsewhere. Most companies will list their trials, and that is a way for you as a patient to get onto that website and actually to see what types of trials are being done in a condition that you're interested in. So Stan, I think the process by which we've met each other and started working on this trial is actually a fascinating one. And I think that would be great if you can share it with the audience as to how your scientific discoveries have gotten us here now.
Absolutely. I think the first thing I should say is that the process we're talking about has taken more than 15 years. So everyone needs to understand that the path to a drug being in the local drugstore is an extraordinarily long one. And we're very fortunate in this work because sometimes it doesn't happen in any given investigator's lifetime. It might take 2 or 3 series of investigators or many to get to the point of clinical drug development. But this one is very much of a fairy tale from my perspective because it's turned out well so far. When I started this work, I really wanted to understand what is happening in the tissues when somebody develops lymphedema. I think to have an effective drug, you need to be able to make the diseased tissues able to coexist with the abnormality. That's what really clears up disease. And sometimes it's not that way. For antibiotics, you just want to kill the bugs. But for many chronic diseases that we treat, we are really helping individuals to overcome the tissue malady that occurs because of some structural or functional problem in the body. In lymphedema, the tissue that becomes diseased is the skin. That's where the lymphatics live. And so my first attempt to follow a path to treatment of lymphedema with drugs was to look at the skin in lymphedema and ask the question, what's happening there? What is the skin doing in response to this chronic edema? So I developed an animal model in the mouse where we could harvest skin after we created lymphedema that's very similar to the type that occurs after cancer surgeries or after traumas. We disrupt the lymphatics and we allow the skin to develop lymphedema. And when we harvest the skin and study the molecular underpinnings of the disease, we look at the blueprint that the tissue is following to exist at that moment in time in the setting of edema, and we compare that blueprint to the blueprint of a healthy segment of skin, we were very surprised to learn that well over 90% of that blueprint was occupied by a response to inflammation. This was a very new concept at the time because in that era, people talked about lymphedema as being a plumbing problem, not an inflammatory disease. So having gotten that clue, we began to think that we could treat the inflammation. We treat many inflammatory disorders with medications, and we can reverse them. So we began to look at drugs that would reverse the specific pattern of inflammation that we were seeing in the tissues. And initially, by treating these lymphedema mice, not only did the lymphedema go away, but much to our delight, so did the structural changes in the tissues. Now, in lymphedema, we think of the disease ultimately as becoming a structurally irreversible disease. Once you get to a certain point with lymphedema, historically, the belief was there was no going back. But here we had evidence that, in fact, we did go back. In fact, we rendered the skin of these lymphedema mice to be completely normal, or as close to normal as we could judge. So with that very exciting observation, we decided to move on to look at the same concept in people. Fortunately, that initial drug that we were looking at was able to be repurposed from a drug that was already marketed and approved by the FDA. So we were able to treat about 120 patients with lymphedema. Part of that study was also a placebo-controlled trial, as we've talked about earlier, where we compared the absence of the drug or a placebo for the drug to what happened with the drug. We found that the drug, in fact, was effective. The placebo was not. And I want to share with you a couple of pictures, those of you that are watching, to show you what we saw in that initial study. So we'll look on the next slide. We decided to look at the thickness of the skin with a caliper in order to measure the impact of the drug. Now, what happens in lymphedema is that because of the accumulation of fluid And proteins in the tissues and in the spaces around the cells, the skin gradually becomes thicker and thicker. Therapists sometimes refer to this as fibrosis. And an element of fibrosis is there, but a lot of it is just the disease in the skin. So what you're looking at here is the same patient before exposure to the drug and after 4 months of treatment with the drug. I've noted on the slide that the normal minimal thickness of the skin that you can pick up with a caliper is usually about 1 to 2 millimeters on the forearm, which is what you're looking at here. You can see that this patient, prior to receiving the drug, had a thickness of 35 millimeters, so 15 to 20 times the normal thickness of the skin. After 4 months of receiving the drug, the thickness is effectively normal or just above normal. So a normal thickness would be 1 to 2 millimeters, and this particular measurement was 3 millimeters. I'd like to show you on the next slide what that translates into under the microscope. So what you're looking at here is a segment of skin that we harvested with a simple biopsy. On the upper panel is the skin before the drug was administered, and the second sample of skin was obtained 4 months later. I have post-processed these pictures in Photoshop so you can more clearly see on your computer screens what we're talking about. The red squiggly material is collagen. Collagen is a normal substance found in the underlayer of the skin. And what you'll notice is that as we transition from the before to the after, about 60% or more of the black area goes away. Now that black area is the area around the cells and around the collagen fibers, and it contains all of the water and all of the protein that accumulates in the tissues because of lymphedema. So you can see that the way that the skin is becoming more elastic is that the ability to clear this material by reducing the inflammation is enhanced. And in fact, we're basically getting rid of the edema and making the skin more elastic by inference, by getting the lymphatics to function more normally. So these were very exciting results. We're actually currently processing all of this data to publish as a forerunner study to the one that we're talking about today, and it really was the human proof of concept for the path that we've been following. Now, this trial confirmed a lot of things for us. It told us that what we saw in the mice was similar to what we might expect in humans, and that is a big step, of course, in trying to treat human disease. But we also wanted to have a firmer grasp on the mechanisms surrounding what we were doing. And this becomes a very lengthy scientific story, which I'll try to put into synopsis form here in the next couple of minutes. But basically, we went back to the mouse model and began to ask ourselves how specifically the drug that we were using was able to accomplish this. And this led us to the discovery that the molecule that is being generated in the skin that is impairing lymphatic function is one that is a known inflammatory mediator. It's called LTB4. And we were able to determine that when LTB4 levels rise, they basically impair the mechanisms that the body uses to repair the lymphatics directly. So when anybody has a surgery or a trauma or an infection and the lymphatics become damaged, the body has machinery to fix that damage and prevent lymphedema from occurring. And what happens in people who do develop lymphedema is that machinery becomes broken. And the broken piece is the elevation of the levels of LTB4. So we were able, with that information, to determine that patients who have lymphedema have very high levels of LTB4 in the blood compared to healthy individuals. And we were able to determine that by specifically and very directly blocking the production of LTB4, that we could actually accomplish exactly what we did with the prior drug and actually accomplished it in a more effective manner. So the drug that we used in those animal studies is Ubenimex, which is actually the subject of the clinical trial that we are now undertaking in human patients. So to summarize, we had basically 3 steps leading up to this clinical trial. Step number 1 was to identify lymphedema as an inflammatory disease. Step number 2 was to identify the fact that lymphedema, this inflammatory disease, could be reversed by blocking the inflammation. And then step number 3 was to identify the very specific mechanism responsible for that unique pattern of inflammation that we see in lymphedema, allowing us to identify a drug that would be potentially able to accomplish this goal in human subjects with lymphedema.
So I think that's a great story, Stan. A couple things really are pretty striking. I think one is that the concept that you had a number of years ago, that it's a skin structural problem as opposed to a plumbing problem, I think is quite unique. And also really the amount of animal work and the human observation and then the animal work and then coming back to the human, I think is a very common link that we see. And also I think just the amount of time and thinking and investigation and different types of experiments that need to be done in order to get you to where you are today. So I think those are actually some really interesting points, I think, for us to recognize.
Thank you. So we'll have some time to field any questions that you might have about the science once we finish the presentation. But now, Joanne, I'd like to turn things back over to you so that you can tell us a bit about the specific ultra trial that we've designed to test the ability of Ubenimex to help people with lymphedema.
Sure. Let me go back to a few slides. So as Stan has mentioned, a lot of his work over the past 15, 18 years really has brought us to the point where we feel we're ready to test this concept in people with a more targeted approach. And so the drug Ubenimex we are using in this phase 2 study. Okay, we coined an acronym, so we call the study ULTRA, and you'll see there on the slide where we were able to pull those letters out. Partly it's because the title is actually a little bit long for us to be referring to all the time. And really, as I said earlier, this is a phase 2 study, so we're looking to look at safety and efficacy in patients with lymphedema. And we decided to study patients with leg lymphedema. Not to say that this drug we think would not work for other types of lymphedema, but what we wanted to do was to very carefully look and design some tools to look at leg lymphedema, and then we'd use this really as a branching off point in order to study different types of lymphedema, different body parts. Okay. For this particular study, we have 2 what we call treatment arms. One is Ubenimex, or the active drug, and the other one is a placebo. So the placebo looks identical to Ubenimex, same capsules, same capsule shells, but it does not have the active drug in it. And the importance of this is because, as you all know, lymphedema can wax and wane, and it can wax and wane with your diet, your exercise, your regimen of self-care, how much you use compression. And what we're doing by doing this blinded is to try to keep all of those variables sort of on a level playing field. So by us not knowing, by you as a patient, if you're participating in the study not knowing, and really no one knowing at that point, it keeps everyone unbiased in terms of assessing any changes. We've also designed the study so that it's randomized 1-to-1. In other words, it's essentially the flip of a coin as to whether you get Ubenimex or placebo. The code is known, but it's not known at the time that we're conducting the study. So all the assessments that are done during the course of the study, measuring skin thickness or limb volume, are done without us knowing whether you're getting the active drug or not. So there's no way that we can bias that. You can imagine, for instance, if Dr. Rockson knew that you were getting active drug and he thought that it really worked, he might actually pinch a little harder or do something a little different, not in a conscious way, but in an unconscious way. And keeping things blinded and randomized actually minimizes the bias that any of us might have. And therefore, when we get a result, we're more confident that it's actually a result of the drug treatment. For the ULTRA study, we actually have 4 sites. 3 of them are located in the United States. We have one at Stanford, California. We have one in Ohio and one in Florida, and one site also in Australia. Next slide. So the basic study design is we have what we call a screening period. This is 3 to 4 visits at which we make sure that your history is understood, make sure that you qualify for the study. And then the treatment period starts. This particular study involves 24 weeks of treatment. And then there are about 7 study visits that are sprinkled in there to make sure that things are going well, to evaluate any side effects, to give new drug. And then at the end of that 24 weeks, the drug is stopped and we have a follow-up for 4 weeks. So there are a total of 11 to 12 clinic visits. In this particular study, most visits are 2 to 3 hours just to get all the assessments done. A few visits are longer. There are some things like, for instance, we need to do pharmacokinetics or make sure that we understand, how much drug gets into the body at various points in time, and that turns out to be a long day. But as you can imagine, those types of things are very important for us to understand in patients with lymphedema. So we carefully look at what information we need to get out of the study and also carefully consider the time and inconvenience that it takes for you as a patient to participate and really have come out with this study protocol. The duration that any individual patient who participates in the study is involved is about 9 months from beginning the first day of screening to the last day of the follow-up. And important to note that as IGER as a sponsor of the study is covering the cost of all the study visits, any study procedures, any drug or anything that's done as part of the study. Next slide. The types of outcomes that we'll look at in this study are things that are related to efficacy. In other words, does it work? Or safety. Does it appear to be safe? So as Stan has already mentioned, we're looking at skin thickness. We'll look at leg volume by doing serial tape measures on the legs. We'll look at bioimpedance, which is a tool that's more commonly being used to look at the amount of water within the limb. And then we also have a lymphedema questionnaire. We know that a lot of the symptoms and things such as pain, tightness in the limb are very important, and we want to assess those. So we've developed a systematic way to gather that. So all of those things help us look at efficacy. On the safety side, we'll look at laboratory values. So you'll have blood drawn at periodic times during the study. We'll capture symptoms, ask you how you might feel, and also to do a physical exam at various points in the study. And things such as EKG, which I think most people have had at some point. Next slide. Very basically, the entry criteria is that we're looking for patients with leg lymphedema. It can be secondary, so secondary to another inciting incident. Some patients may have had surgical removal of lymph nodes due to cancer or radiation. That would be secondary lymphedema. We've also recently modified the protocol, changed the protocol, so that we will allow some patients with primary leg lymphedema. We want to be studying, at this point, the patients with primary lymphedema, which is not of congenital onset. Okay? For this study, we're looking at enrolling patients between the ages of 18 and 75. And we want all patients to be able to walk, and we're allowing a walking aid, such as a cane. So those are very basic entry criteria. If this study is of interest to you, we'll have a website in a moment where you can give us some basic information and someone can contact you. And then they will work with you to ask you some more questions. And then ultimately, if it looks like you still might qualify, then we'd send you to a study center, and then they can do a more complete evaluation. So I think that gives you some basic outlines of what we're looking at in this study. And the next slide will show what we have set up as a website. It's called www.igerle.com, LE for lymphedema. And you'll see the landing page there. And if you click on that, then you can enter answers to some kind of basic questions and also enter your contact information and name if you give us permission to contact you. And then there can be a brief discussion over the phone, and then we can refer you hopefully to a site that's close to you. Okay. With that, perhaps we should go to some questions?
Sure.
Okay. So one of the questions that someone has written in, and maybe you can address this, Stan, would this therapeutic pathway be effective for primary and or secondary lymphedema? Maybe you can give us some thoughts about that.
In principle, yes. I will say that in the 120 patient study that we did as the forerunner to ULTRA, we did include both primary and secondary lymphedema. The lymphedema patients that were entered there had lymphedema of variable duration, anywhere from 6 months to 6 decades. And the secondary lymphedema was of a variety of types. We did have primary lymphedema, and we think that the mechanism that we are looking at is a very unifying mechanism in the body in which the skin responds to chronic edema. So that in principle, in my mind, all lymphedema should respond to the drug. Now, the one question that we have, which will need to be addressed as we move on, is whether the inherited lymphedemas that occur as a result of faulty lymphatic development will have the capacity to respond. Because remember, the drug basically unblocks the repair mechanism. But if the problem is not one of faulty repair, but rather one of faulty development, we don't know if the drug will be useful. It certainly might be, but we'll need to ask that question in the future. That's the reason that we've excluded congenital onset lymphedema from the current study, meaning any individual, no matter how old they are, if the lymphedema was present when they were born, they probably should not be in this first study because we don't exactly know how to predict how that will be affected, but we'll be certainly asking that question in the future. So let's move on to the next question. We've been asked, Joanne, what lymphedema questionnaire are you using for the study?
Yes, so we are using a questionnaire that we've named the prelim leg, but it really is what's called the LymQual that was developed by Dr. Von Keeley a number of years ago. Some of you may be familiar with that particular instrument. That is actually a fairly lengthy questionnaire, and what we did was to work to shorten it and make it a little bit easier to administer. But all the basic types of questions we've grouped, which are really derived from all of that previous work. And so the way that we developed that questionnaire was to look at that questionnaire and also when we spoke, Stan, a couple of years ago, we gathered just anecdotal information that you were having from your patients in terms of the types of symptoms that they experienced and what changes they felt that they might have seen with the other trial that you did. And we incorporated all of that into the questionnaire. We're obviously very aware that the symptoms that patients feel is incredibly important to this. So it's really a very important tool for us to understand if a drug is having a positive effect.
Okay, another question, Joanne, is whether all patients in the study get active drug at one point.
That's a good question. In this particular study, we are randomizing it so that the patient gets either active drug, Ubenimex, or placebo. But we do not have a provision for all patients to get active drug at this point. This is really still the first proof of concept study for Ubenimex as an investigational medicine in lymphedema. Obviously, if the study is positive, then that gives us a lot of reason to move forward and design other studies. And sometimes, having some positive data in terms of safety and efficacy is a good reason for us to allow people who have finished the study to continue for some time. So the specific answer to that question is that the ULTRA study is designed so that patients either get randomized to active drug or placebo, but not all patients will get active drug at some point.
Great. Next question. Why should a patient consider signing up to participate?
I think that's a good question. And I think part of it is, I think when you step back, I think especially with the way that you framed the initial thought about lymphedema and what might cause it and how you might intervene, it's pretty clear that it takes a lot of people and a lot of effort to develop a drug. And I think as I've come to understand the unmet needs in lymphedema, there's a lot of frustration, a lot of suffering that's out there. Really, the reason to consider entering a trial is to change that. Drugs are developed with a systematic approach, like doing many clinical trials. And I think if the lymphedema patient community starts to understand that and appreciate that, then enrolling in a trial is one way to change the fact that there really are no drugs right now. We certainly hope that anyone who signs up for a trial is going to benefit from it. That may or may not happen, but it will benefit the entire community in that respect. And there, in some cases, I've been aware with other trials that I've run, that sometimes people's care is not as consistent, and some people sign up for trials because they know they'll be seen by a top-notch team and be seen regularly. And that is a value to them as well.
I agree with all those things. And if I could just add a personal comment, I know from working with the lymphedema community over the last 25 years that there has been an extraordinary level of frustration at the non-responsiveness of the system to provide treatments. And this is an extraordinary glimmer of hope for the lymphedema community. We now have a drug that is potentially very useful and may be a big breakthrough. And everything we learn from this study, including the activity surrounding the placebo patients, will drive that agenda forward and make these treatments more and more successful. So this is a personal investment in the future for all lymphedema patients. It's the difference between taking your bonus and buying a car today or putting it in the bank and deciding that one day you'll be able to buy cars every 5 years. I think it's an important investment for everybody who can participate in the lymphedema community. So I've been asked another question, Joanne. Somebody lives in Illinois. Can I participate in Ohio?
That's certainly very possible. We have to make sure that you would qualify for the study, and so referring you to the Ohio site and having them evaluate you, and then we just have to figure out the logistics. We are aware that there's some people who live a bit of a distance from a study site, but we have some things that we hope can help make that a little easier. So we would encourage that. If there are people who don't live too close, as long as you're willing to invest the time and the effort into the trial, we'd support that as much as possible.
And somebody asks, do I have to be in the trial for the entire study?
Well, the idea of the study is that we would be able to test how you do, and we've developed a study protocol very specifically to look at the effects of 6-month treatment. So the idea is that if you go to sign up for the study, we really do want you to participate for the entire study, because then we can get the most information out of that. We recognize if something happens, as I said earlier, participation is always voluntary, so that you do have the right to withdraw at any time. But we do think it's important that we, at the beginning, make sure that you understand really the commitment for the study. And the most information we get out of it is someone participates fully in the study, does all the study procedures, comes to all the study visits, fills out the questionnaires. That gives us the most amount of information from your participation. So that's what we would encourage.
This is an important question. Somebody wants to know, why can't I know if I'm on active drug or placebo?
Well, I think that's a very fair question. And I think that relates to what we talked earlier about possible sources of bias. I can imagine if I'm a patient and I know that I'm active drug, I may just be a little bit more diligent about wearing my compression garments. I may be a little bit more diligent about my self-care, about my manual lymphatic drainage. And those things could have an effect apart from the effect of the drug to make it look like my lymphedema is better. And so the reason why this is done in a blinded fashion, so you don't know, we actually don't know, no one at IGER knows at this point, and actually, Stan, for the study, for the patients that you've involved, you don't know either.
Correct.
Right? Nor does your coordinator, right?
Correct.
So everyone is blinded, and we think that's very important so that we can have an unbiased view of anything that changes in the study. In the end, what we do when everyone is completed with the study, then we do what we call unblinding, and then we figure out who was on what, and we look to see what the results are. But during the course of the study, we think it's very important for a study like this that it's blinded. And also, we mentioned earlier, we capture symptoms from patients. If you knew that you were on an active study drug, you may say, oh, well, I don't think that's related to my lymphedema. But it may be. So I think we want to be very unbiased with this, and one of the ways that we do that is to keep it blinded.
I think it's maybe fair to add that pretty much any medication that anybody takes today in the audience that is listening at one point was tested in a placebo-controlled fashion. That basically is the tried and true mechanism whereby all new drug and device development occurs. It's not something that we're imposing specifically to the lymphedema community. We're using what is commonly employed in medicine and in medical investigation to create new treatments.
Right, right. And I think that's really surprising, I think, to many people. Until I started working in industry, I didn't realize that. But if you pull any drug label, that really long piece of paper with a very fine print that you get when you get your medicines from the pharmacist, and you look carefully, you'll see that there is a lot of information about clinical trials. And those are all trials that patients have participated in to help develop that drug. And so I think we all, at some point, take medicines. We're all patients at some point. And I think, that observation is really to say, we should contribute in our own way to developing new drugs. Okay. Stan, so one question that someone in the audience has asked, would this drug be used for life or just until symptoms subside?
It's an excellent question, one that I harbor myself, and I have to say, unfortunately, I don't know the answer yet. I think there are a couple of ways of thinking about this. It's possible that if the disease really is a failure for the lymphatic system to repair itself, that if we give the system a boost over a period of time and it does repair itself, that repair might be self-sustaining and there might be a time in which treatment is no longer necessary. Alternatively, it might be a system that breaks down and if it isn't continuously helped, or it might be that we will eventually evolve a treatment strategy that has to do with the approach to the disease itself and then to a maintenance phase once the benefit has been attained. And of course, what is still unanswered, and is a very provocative question, is whether at some point in the future, Ubenimex might actually be a way to prevent lymphedema. If you know an individual is at high risk for having faulty lymphatic repair, there might be a time at which the drug could be administered to prevent the process from occurring in the first place.
So I think actually those are great questions, and a lot of those questions would need to be answered by specific studies.
Exactly.
Right? And so really the ULTRA trial right now is really sticking the toe in the water to see if it looks like it works. And then there's a number of different directions that we can take it in terms of different stages of disease, in terms of disease of different body parts, arm, leg,
Right.
trunk,
Right.
whatever. So this is kind of the springboard
Right.
for us moving forward.
Right. So what does it mean for a patient if he or she does not qualify for the trial? Will that person ever get access to the drug?
Well, if you don't happen to qualify for the trial right now, it is still possible that you might qualify for a future trial. And on the IGERLE.com website, if you enter your name, then we'll have it on file and we'll be able to contact you if it looks like you may qualify. For instance, if you have arm lymphedema, right now we're recruiting for a study with leg lymphedema. At some point later, we might very well do an arm lymphedema study, and we could contact you.
Right.
So while the drug is being developed, the access to the drug is through a study protocol. At which point, if we're so lucky as to have the information could be compelling enough and submit it to the FDA and have them approve it, then it would be available, and your doctor would be able to prescribe it.
Just to say it another way, from my perspective, if you don't qualify for a particular trial, it's not because your disease is not amenable to the drug. It's because you don't fit the template of the specific situation that we're trying to study in that particular trial. We need the individuals in a trial to be as similar to one another as possible so that we can group their responses to the drug and look at the group behavior. And if there are outliers to that group phenomenon, it's going to hamper our ability to do the analysis.
Okay. Stan, here's a question from someone in the audience. If biopsies are taken from the leg as part of the trial, what safeguards are present to ensure that especially placebo patients do not experience worsening of their conditioning?
That's a great question. So I want to help everybody listening know that I've been doing skin biopsies and lymphedema for most of the 18 years that I've been doing this work. And I can say out of the many hundreds of people that I've biopsied, I have never had a situation of worsening lymphedema or infection or any other complication that would be addressable to the biopsy itself. In my hands and in the hands of the current clinical protocol that I helped to create, we do use a short exposure to prophylactic antibiotics during 1 or 2 days after the biopsy to completely ensure that there is no problem, but strictly speaking, we do these biopsies under sterile conditions, and we do it with the knowledge that lymphedema is the underlying substrate. But I can tell you that while I can't give a warranty that there will never be a problem, I have never yet encountered one.
So Stan, here's a question. Is there a genetic link or predisposition to lymphedema or elevated levels of LTB4?
So that's a two-part question with a two-part answer. The genetic question would have the answer yes in certain cases. We have now identified collectively in the practice of lymphedema probably 12 or so individual genes that are known to mutate in certain ways that will produce a variety of forms of primary lymphedema. Most of them are congenital in nature, but not all of them. The 8 or 10 or 12 genes that we've identified probably represent no more than 25% of the whole primary lymphedema population, and probably a lot less than that. So there are more questions than there are answers at this point in time. We believe that all the primary lymphedemas are on some form of a genetic basis because they are unprovoked by outside circumstances. They simply occur within the body at specified periods in the life cycle of the individual. About the LTB4, to my knowledge, nobody has ever identified a genetic condition in which there is a genetic predisposition to overproduce LTB4. That's a very finely regulated inflammatory mechanism within the body that has lots of checks and balances, and I don't know that any imbalance to that degree would even necessarily be compatible with a normal life. We know that LTB4 expression plays a role in a wide variety of diseases beyond lymphedema. We're just focused on the lymphedema component. But we think what's happening in lymphedema is that when the lymphatic circulation becomes faulty, the circulation of inflammatory cells into and out of the lymphedema tissue is faulty because of the lymphatics and their inability to carry those cells. And it is that process that leads to the local overproduction of LTB4 that in turn leads to the complete expression of the disease. So it really is a manifestation of the primary problem that is present within the lymphatics themselves. So another question, Joanne, can Canadians participate in Ohio? And a second part of that question, will you open study centers in Europe?
So for the first question, yes, we'd be very happy if there are Canadians participating in the study. The fact that you're Canadian is not an issue for us. We would just have to make sure that you're in contact with the site in Ohio, that you qualify for the study, and you arrange the logistics with them. Currently, as I said earlier, we have 3 study sites in the United States and 1 study site in Australia. We certainly are aware that there are many patients in Europe and there are many good centers in Europe. Currently, we're enrolling at these 4 sites, and then we would look to expand beyond that, with subsequent studies. So our current plan is not to open study sites in Europe just yet, but it is something that we're thinking about in terms of expanding the geographic reach of the study.
So a good follow-up question, how long will the ULTRA study last?
Well, we're still enrolling right now, and we expect to be enrolling for a number of more months. And obviously, how long we enroll depends on how quickly patients come forward. We're looking to enroll about 40, 45 patients. So there's certainly still time to contact the study center and find out about the study.
If someone has upper limb lymphedema, can that individual give information for future studies?
Oh, definitely. And that's the reason why we set up the IGERLE.com website. So that is, a patient who may or may not qualify for the study can enter their information in, and then we will have it as a database. And in the future, if it looks like there is a study that you may qualify for, we will reach back out and contact you.
And so follow-up question to that, an individual asks, if I have lymphedema in my legs and arms, do I qualify?
Currently, the ULTRA study is written so that we're just looking at leg lymphedema. And so you would not qualify for this particular study. But again, that is, having lymphedema in a more widespread fashion is something that in the future we may be interested in studying. So you can certainly go to igerle.com and enter your information there. So Stan, another question about skin biopsies. Where are the skin biopsies taken on the body?
So for the purpose of this study, of course, they will be in the leg because the leg is the involved segment. We try to do this on the inner side of the calf. So it's below the knee, more or less midway between the knee and the ankle, on the part of the calf that would be most interior to the body, so the least visible. And that's been the place that we have typically sampled the skin. I want to be sure that everybody on this webcast understands that the procedure of the skin biopsy is exactly what would occur in a dermatologist's office. If somebody has an unidentified skin rash and the doctor cannot make a diagnosis by simple observation, the standard technique is to do one of these skin biopsies to look at the skin under the microscope. It really is considered within the scope of clinical medicine to be a pretty trivial intervention. It is effectively painless. Of course, we have to use a tiny dose of local anesthetic before the biopsy is taken, but the whole procedure takes under 3 minutes, and it's quite simple and really, in my opinion, shouldn't be a cause for undue alarm. So, next question. Although there are risks associated with drug trials, what information can you provide to ease my family's fear of study participation?
That's a good question. Oftentimes, I think a family member or members can actually go with you to the study visit and ask questions of you, Stan, or the coordinators to make sure that you understand fully what's involved in the study and what risks you have. I think some of the patient protections that we talked about earlier, the fact that this study and really all the studies that IGER sponsors are reviewed by an institutional review board. Stanford has an IRB that the study has been reviewed at. The FDA has oversight as well. So there's a number of protections that may not be immediately obvious to you in terms of other people and other agencies looking at this carefully. Right. As part of us filing the information with FDA, for instance, we are obligated to periodically pull safety information together and submit it to the FDA. Safety issues of a certain nature are immediately reported to them. So there's a number of different mechanisms by which we will be looking at the safety, and others, such as the IRB and FDA, will look at it. Right. But I think it starts with including, if appropriate, your family members coming with you to the study visit and making sure that all of your questions are answered about the study and making sure that you understand the risks. I think that's very important, the process to make sure that you understand what is involved and what is undertaken and that you're comfortable with it is incredibly important here.
If I can add to that, just the experience for individuals who come to our site here at Stanford. When we go through the initial screening process with an individual before they ever are in the trial, we come to a certain point where we realize that that individual meets all of our inclusion criteria and they are sufficiently interested to consider the trial. We always provide a written copy of the informed consent document for their review, even prior to the first visit, so that they know as much as we can provide information about safety and risks and alternatives. All of that is required to be spelled out in the informed consent document, and we do it in great detail and in language that should be really able to be processed by anybody who reads it. But then if there are questions beyond whatever the document is able to provide, then we're happy to answer those in question in person as well.
Right. And I think, unfortunately, as a result of providing all the information, the informed consent document is typically very long.
Yes, it is.
So it appears a bit daunting to patients, but the reason for that is because it is really an element of disclosure. Yes. So I think it is, and it may be a bit overwhelming. So, you as a patient, if you're thinking about participating, it's fine to say, I have to think about this and I have to talk to family members and I want to come back. That there's no problem with that. We want to make sure that anyone who participates in any study really understands what is involved. And if at some point you decide it's not correct, not right for you, then that's fine. I mean, we very much appreciate that there's some things that may feel right for some people and not for others. So this is in no way meant to be coercive in any way, but just to make sure that people are informed and can make a good decision for themselves.
Right. So we have had a couple of interrelated questions. There are people on the webcast who have had a variety of the microsurgical techniques for lymphedema, either lymphovenous bypass or a lymph node transfer procedure in the past, and there's some curiosity about whether they might qualify for the study.
Yeah, so I know that, Stan, you and I had discussed that early on, and we certainly are encouraging patients who have had previous surgical procedures to come forward. What we're asking is that patients would have had it done at least a year ago. They still have residual disease, and they're relatively stable from whatever things have been done in the past. So we're certainly aware that there are a number of surgical procedures that are available these days, and they provide significant benefit, but they may not actually either work in everyone, or people may still have a significant amount of residual disease. So again, that's a group of patients in whom we think that a drug such as Ubenimex could have a very positive impact. So that's why we've included them within the patient population within the study.
Right. Will patients have access to their own clinical study data if they participate?
So certainly all of the study data that we gather is available, to you as the investigator. And I think sometimes, patients do find that having that close look at their disease is actually helpful in and of itself. So I think, yes, the answer to that is yes.
Great.
And a question for you, Stan. I have cellulitis in my legs with my lymphedema. Do I qualify?
That is a question that has a variable answer. Cellulitis in the past is not a contraindication, and we actually think indirectly that if Ubenimex is successful, one of the attributes of the treatment response will be a reduction in the likelihood of cellulitis in the future. However, we don't want active infection or antibiotic exposure during the enrollment period itself. So the protocol does specify a certain period of time since the last exposure to antibiotics, and if an infection occurs during the course of the study, it may require that the study be terminated at that point, depending on the circumstances. But anybody who has a history of cellulitis should not feel that they need to hesitate about contacting IGER or considering participation if they're otherwise interested. So, Joanne, someone asks, can I have access to my genetic testing data?
So as part of this study, we have included that we would like to have patients consent to collect a genetic sample. We currently are not analyzing those right now. We're kind of banking those, and if some questions come up in the future, then that would give us the permission to test that. So currently we're not testing the presence of specific mutations, for instance, and so therefore there's really nothing that we would be disclosing at this point.
Right, and that genetic testing would have a lot to do with the metabolic pathways related to LTB4 rather than to the mechanism whereby the lymphedema developed in the first place,
right,
in all likelihood.
Right.
So it wouldn't shed a lot of clinical information, even if it were.
Right. I think a lot of that is really much more on the research side that starts to develop some of the ideas, for instance, that you shared earlier, and it is probably not particularly ready for your physician to use in terms of their clinical care for you, and so that's the reason why it's purely done on a research basis.
When will the drug come to market?
Well, everyone would want to know that. I think there's a number of things that have to happen. We have to show that it's safe and effective, and this is only the first study within a lymphedema population in which we have the opportunity to show that. As I said earlier, there are other studies in which we would have to do in order to have really compelling information as a package to take this to FDA and other health authorities to have it approved. So if this drug works, it would still take a number of years for us to complete the trials and gather all the information in order to bring it forward. And I think that's part of what you as patients should appreciate, that for any drug to get to the market takes a lot of effort by a lot of different people, all the way from the first light bulb going off in your head stand to working with the animal models to bring it back to the clinic and then doing some expanded studies. Unfortunately, it takes a lot of time because we need to gather a lot of detailed information to make sure that this is safe and effective.
Right.
So.
Well, for those of you on the webcast, I think we've come to the end of our allotted time. So I just want to take a moment to thank everyone for their active participation, either listening or asking your specific questions, because I think those have been very helpful to the audience at large. And I want to thank Joanne for our partnership.
Thanks very much.
And for all of the exciting news, I think, that we have for the lymphedema community, that there is, I believe, real hope on the horizon.
Thanks.